Okay, this video is the second part for CEP 636, week 11, the fatigue process. And I believe that we've stopped at slide number 30. I'm continuing from slide number 30. So in regards to slide number 30, I just want to say about the first point. The first point is talking about information going into the brain, right? Because we're talking about an afferent with an A. And then we see in the next 2 points that everything that uh, um, is influenced is also influenced by the type of information that you're giving and not just the idea that you are giving information. Okay? Um, so you see, for instance, motivation, information that indicates motivation, information that indicates mindset, and the nature of activity influence the outcome or central fatigue as much as other things. Then we can talk in slide 31. First of all, we're talking about maximal voluntary contraction, MVC. We see that the second point is talking about a one-directional process. And then we see that the third point is emphasizing the accumulation of information, meaning that central fatigue is not a one-timer, so to speak, and that's it. It's part of accumulation of information. There's a certain threshold that has to be reached due to the amount of information in order to have central fatigue. Then in slide 32, we can see that the first point is talking about getting faster and more accurate work to rate regulation, and better regulation is definitely part of better function, right? Then the second point, I wrote a remark, I wrote the pyruvate junction. So obviously I want you to think on how this loop of information, of feedback, is influenced by the pyruvate junction, which you definitely should know by now. Then we see I wrote regarding the third point on slide 32 that that this is also metabolism dependent. So here's the connection between potassium and regulation and metabolism. Then in regards to slide 33, where we have the idea that central fatigue is influenced by the firing rate and the alpha motor neuron number, and of course the combination of both. In regards to slide 34, we can talk about the ATPase pumps, about the ion pumps in the membranes of the cell, and of course membrane permeability. Then the next slide obviously is all about hormones, and as I always say, if there are two substances that have been given superpowers in the body, it's enzymes and hormones. So obviously this extrinsic influence is very powerful if you utilize a hormonal pathway. Then in regards to high intensity exercise, this is the first place where I saw I made a mistake and I wrote lactate acid. I think it appears twice or three times again in the presentation, so I would like to apologize. I've already corrected it, but of course it's lactic acid and not lactate acid. Then in regards to slide 39, we're talking about phosphocreatine or creatine phosphate. This whole slide should lead you to think that mechanical direct or indirect factors are more important in this case than the chemical. And I'm not saying the chemical is not important, but obviously the chapter, the way they wrote it, and the vision or the principle that they're trying to slowly get us to understand is that fatigue is not just chemical, but also the actual physical inability to produce power, and so that is more of a mechanical standpoint. Um, then in slide 40, we see once again that balance that always exists regarding the body of delivery versus um, utilization. So it's not enough to deliver, it's not enough to extract, so to speak. You also have to have the ability to utilize what you have delivered, and that's what we see basically in the background of slide 42. 40, sorry, slide 40. Then we can see in the second point of slide 41 that ADP is a signal and a second messenger, meaning it's a regulator. It's a very strong and important regulator. And then in slide 3, we see that we need ADP to be available to create ATP. That's what the whole point is talking about. There's a conflict here, a very short, well-localized conflict between the need for ADP as a signal, second messenger, and regulator, and as one of the components in ATP. Then if we go to slide 43, we can see how pH can impair anaerobic enzymes as a safety mechanism of sort. An interesting idea within itself. Then the next slide talks about EC coupling, excitation contracting coupling, the idea of producing function through a process that includes neural or the nervous system, and of course the mechanical aspect of the muscle. In regards to slide 45, the third point, the last point, we can say that no neural input equals no mechanical change equals no power. And that is, of course, the failure, the possible failure of the EC coupling. Going to slide 47, we see that, and this is not new, that we need a conformation change, which once again takes us to the principles of anatomy and physiology, or structure and function, how structure influences function. Then in regards to slide 48, the first point, we're talking about the idea of being more effective. And then in the second point, we can talk about some kind of delay, actual physical delay of the process. And then we can talk about recruiting muscles that either become stop being an option for a while or are delayed, and that means that you will produce less power and less work. In regards to slide 49, just addressing the third point, of course we have here a gradient change, and gradients are driving forces of the body, of physiology. So if you have a change in the gradient, you immediately have a change in function. I'm going to say it again. If you have a change in a gradient, you immediately have a change in function. And then we see in slide 50 that basically we have a few examples where the changes to the gradient are too great, meaning the body is not able to cope with this, and this usually means that you've just lost regulation or supervision or balance, and that is never a good thing. So yeah, so as I said, and this is in regard to slide 51, gradients are forces of change, and they are driving forces of the system. So when you play with them or change them, or they lose regulation or supervision or balance, you have a problem in the system. In regards to slide 52, we can talk about DHPR being a calcium channel, and we know that calcium is used as a biophysical weight in order to rotate actin in such a way that myosin can actually connect to it and create those cross bridges. Then we see that chloride is a calcium release regulator. Once again, more and more layers of regulation and safety, so things don't happen at random usually. Then in regards to slide 53, we see a very unique dosage relationship, which is absolute, and we don't see many of those, so that is relatively rare. Then we can start talking about the charge sensor, which is an interesting idea. So the two next slides, regarding the two first slides, regarding that subtitle of the charge sensor, is the idea of the accumulation of electric potential in order to activate the muscles. And in this case, we're talking about a charge sensor in the sense of creating a sort of biological battery in order to operate the muscle or charge the muscle with energy. Then, if we talk about slide 58, and we're talking about RYR1, that is a calcium release channel of the SR. And then the third point on that slide 58 once again talks about the idea of 3D conformation, the allosteric or 3D change in arrangement of a protein or a complex as a switch between situations. So if you do not achieve that change, you will not switch from one situation to another, from one result to another, from one option of certain function to another. And then we can talk about the second point in slide 59 that says, thus the SR calcium release and removal process have a major role in the production of fatigue, meaning if it happens and how fast is part of possible inability to produce work, hence fatigue. And then the third point on slide 59 talks about a gradient flow relationship. When we're talking about slide number 60, this is an important point always, which we will revisit deeply and essentially in pathophysiology, whereas change to the content of the cell's fluid immediately changes not only function but the chances of surviving. And then of course you're playing with plasticity versus elasticity, and you're making certain areas of the cell more viscous, which tends to resist movement, and the cell is alive, or the more reactions you have in a cell, the more movement of substrates and substances in a cell, you can say that the more that cell is alive. And then I just want to remind you that there's no such there's not really, from a neural standpoint, such a thing as muscle relaxation, rather than the lack of activation. There's no neural impulse that contains the order to relax. It's the lack of uh, uh, neural impulse that does not activate a muscle. There's a difference between the two in the mechanism. Then we see on slide, which number is this? It's hard for me to see. Sorry, 61. That we're talking about a pooling of calcium, accumulation of calcium in a certain area that equals the idea of fatigue. And I believe I already explained to you guys that, and ladies, of course, that inorganic phosphate, PI, is often used as handcuffs of the cell. When you are connected to a phosphate group, you cannot leave the cell. Then we see calcium confined by phosphate. We always want calcium, which is highly reactive, to be connected to something, but we also know that that promotes fatigue because the creation of calcium PI prevents the connection of cross bridges of the sarcomere, and that means that you're not producing power. So by definition, it is an inducer of fatigue. And then we see that ADP, in another role within the cell, is a regulator of the creation of calcium phosphate, meaning that ADP in that sense as well is a regulator of cellular fatigue or muscle fatigue. Now, since ADP is so important and so powerful, you need to regulate ADP as well. And we see that ADP is a phosphocreatine or creatine phosphate dependent. So you see cycles of balances and cascades and feedback loops and safety mechanisms. So there isn't one process that can just go ballistic or crazy. There's always something that is a fail-safe or reliable safety mechanism in case it goes in one direction to the extreme. Now we see that if there are processes that take more time to reverse the situation, and these processes prevent work, then you have what we call more rest or longer rest or a longer recovery process. Then in regards to the to slide 66, I just wrote that I want to remind you that in the periodical table, any particle charged with a +2 and +1, +2 -2 charge tend to be troublemakers and above. The higher the number, the worse they usually are. So obviously calcium is one of them. Furan has a +3. So these guys, so to speak, need to be chaperoned. They need to be regulated. They need to be supervised. They need to be balanced. They need to be confined. Otherwise, they're going to create problems. Then we have slide 67, and we see that glycogen itself acts as a regulator. That is a conclusion from the first two bullets of the slide. Going to slide 69, we can see that half RT is pH independent, meaning that there are other mechanisms and factors that exist to affect it. Then I want to go straight to slide 72. We see calmodulin as another fatigue regulator, a very important one because it regulates the release of calcium from the SR. Slide 73, when we're talking about CMK2, we're talking about a kinase, meaning that it creates a shift from ATP to ADP, meaning that it catabolizes the hydrolysis, the breakdown via water or H2O of ATP, and then you get more ADP. pKa is also a kinase, and of course we can see in the second point that overphosphorylation of R1, RYR1, that calcium release channel leads to fatigue. Then the next two slides, 74 and 75, the only thing that I wrote is that obviously oxygen is highly reactive when it is free, and it doesn't work well for the body. We know about ROSs, and we need to confine them. They are actually one of the major causes of cell injury and aging. Then on slide 77, obviously the two first points lead to less power being produced. And then a major reason is ATP being hydrolyzed. In slide 78, the third point, we see that PI, inorganic phosphate, is also a switch between energetic states. And in the fourth point of that slide, slide 78, we are of course once again talking about affecting the second step, which creates fatigue. As to slide 79, of course, elevated resistance to change of conformation is needed in this case to establish the cross bridges, meaning that this resistance to the change in conformation prevents or lowers the chances or prolongs the time for the sarcomeres cross bridges to be established, and that within itself falls within the definition of fatigue. Then we can see that in high temperatures, less cross bridges are created. Once again, we have loss of power and more fatigue. This could be caused for several reasons. It also depends on what do you define as high temperature, because if we're talking about above 40 degrees Celsius, you're already talking about degradation of proteins, which myosin and actin are of course proteins, and it might affect that. We can talk about expanding of the apparatus of the sarcomere, which means that they might actually move away from each other, and that of course will create more fatigue. In regards to slide number 82, I highlighted the second point, which says that glycogen depletion and SR function are stronger factors of endurance-induced fatigue, meaning that these are two major factors that will induce fatigue under endurance conditions or during endurance tasks. Then in regards to slide 84, of course, when we're talking about the liver and glucose and gluconeogenesis, the recreation of new glucose, we're talking about the Cori cycle within the liver, which is a very important cycle pertaining to performance and competing and existence, actually. Then, in regards to the third point on slide 85, BCAA is branch chain amino acids, and these are essential amino acids which influence protein synthesis, protein creation, and what we call turnover, which is per 24 hours usually. Then, in regards to the next slide, we can talk about glycogen depletion, creating more BCAAs that in turn lead to less aerobic energy, and less aerobic energy is immediately affecting the energetic efficiency of the cell. And that was my last remark. So this is the last video for this topic. If you have any questions, please engage me through email, cell phone, however you would like. Bye. Have a great weekend.